for this session is Dr. Biana Bernshtein from the Reagan Institute of MGH, MIT, and Harvard.
Thanks, Idan. Hi, everyone. Very excited to be here. I apologize for my voice. I've been sick this week, but I found it to be very inappropriate to cancel after we've been postponing this symposium for a year and a half for clear reasons. I really want to thank the organizers for sticking with us and continuing to reschedule until finally we can do this today. So I'm very happy that I get this opportunity to share with you some of my work that I've been doing over the past 5 years, trying to understand how antibodies mediate protection against Shigella infection. And so I'm showing you this kind of weird looking map. The size of each country in this map is disproportionate to its actual size, but it is in proportion to the number of children that died before they reached the age of 5. And you can see how these premature deaths are disproportionately occur across our Earth. Many of these deaths happen due to diarrheal diseases. And you can see in this plot how over the past 20 years, we managed to decrease the number of these deaths from about over 1 million in the year 2000 to about 500,000 in the year 2023. However, we still see all of these deaths occurring even now. And the colors here represent different pathogens that are associated with these diarrheal diseases. So in red is rotavirus, and we have a very good vaccine against rotavirus. So you can see how the number of deaths declined over the past 20 years. But the second leading cause is Shigella here in blue. And you can see how over the past 20 years, there hasn't really been a change in the number of deaths due to Shigella infection. The reason for that is we don't have a licensed vaccine against Shigella, and this is because we don't have a clear correlate of protection. So essentially, we don't understand what protects against Shigella infection. And this is not only a problem that affects low and middle income countries. This also happens in high income countries. So here I'm showing you a collection of cases or outbreaks that were reported over the last 30 years, where strains that were isolated from these outbreaks mostly were multi-drug resistant, suggesting that although today we can treat children with antibiotics in these countries and prevent death, possibly in a couple of years or decades, this will not be a good solution anymore. And this underlines the need for a good vaccine. So usually we use antibodies as correlates of protection. And if we look at this bacteria that is opsonized by antibodies, we can think of 2 different domains that are important for this interaction. One is the Fab domain, and this is the domain that binds to the antigen, heavily studied. In the case of Shigella, we can think of 2 classes of antigens that are of interest. One is protein antigens, and these are these mostly virulence factors that are important for the interaction of Shigella with its host. And the other group of antigens are polysaccharide antigens, or O-specific polysaccharides, or as I will refer to them from now on, OSPs. And these are part of the lipopolysaccharide that is on the surface of Shigella. Now, the other, and if you ask me, much more interesting domain of antibodies is the Fc domain. And this domain is in charge of communicating with the innate immune system. And it does that by binding to Fc receptors that are expressed on the surface of different innate immune cells, and then activating these cells to promote different functions. For example, this monocyte that expresses Fc receptor on its surface will bind to the antibody Fc and promote will phagocytose this bacteria. The same can happen with neutrophils and also complement deposition. And so, as I mentioned, we don't have a vaccine against Shigella, but this is not because we haven't tried. Over the past 40 years, there have been many attempts at developing a Shigella vaccine, but they have continued to fail. And the main obstacle, again, is because we just don't understand what really protects against Shigella infection and severe disease. And to me, this underlined the need to investigate antibody-mediated protection beyond just antibody binding and quantity. And so how do we do that? So this is why we developed this system that allows us to profile antibody responses in an unbiased and broad, high throughput way. And this is a bead-based system where we use fluorescent beads. We conjugate them to an antigen of interest. So you can start thinking that this is probably useful for other pathogens as well. We will then add to these antigen bead-bound antigens any sample that has antibodies in it, so serum, but also breast milk, cord blood, saliva, any sample that has antibodies in it. We will wash away what didn't bind, and now we have this immune complex. So this is antibodies that are bound to an antigen. And the next step would be to ask questions about this immune complex. We can analyze levels of IgGs, IgAs, and IgMs, different subclasses of antibodies. We can ask how these antibodies bind to different Fc receptors, Fc gamma receptors, and Fc alpha receptors. And finally, we can ask how these immune complexes interact with different cells and promote different functions through FCR interactions. And we do that across many different antigens. And because we work with human samples, we need to do this across many different samples because humans are extremely diverse. We build these multidimensional models. I'm sorry, I don't know if you can see this, but we build these multidimensional models of the functional antibody response to any pathogen, but today I'm talking about Shigella, so let's focus on Shigella. And this is the easy part. Now comes the hard part where we need to analyze the data and begin to identify protective signatures within our data. So today, since I don't have a lot of time, I'm going to share one story where we collaborated with the American Army and the Walter Reed Institute of Research, and we collected samples in a military base in Peru that has incredibly high burden of Shigella. So this is a military camp where the recruits come into the camp and we follow them over time. We know when they got infected, and we also collect serum samples so we know exactly when everyone got infected and also which serum sample we have from each individual. And so the way we decided to approach this is by dividing the cohort into two groups. One group, we call them susceptible, and these are individuals that were infected within the first 3 months at camp. The other group, we call them resistant, and this is because they were not infected for at least 3 months, but eventually they did get infected with Shigella. And so we profile their Shigella-specific antibodies. We throw all of this into a model, and we ask which antibody features separate the resistant and the susceptible individuals in this cohort. And the model spit out two main features that are important to separate these two groups. One was OSP-specific antibodies that bind to Fc alpha receptor, and the other was OSP-specific antibodies that activate neutrophils. And both of these were enriched and resistant individuals, and I'm showing you this here, both of these features. And so the question we wanted to ask is this neutrophil phagocytosis that we find is potentially protective, is it mediated by IgA that binds to Fc alpha receptor? And let me try to explain. What I'm showing you here is a neutrophil that we know expresses both Fc alpha receptor and Fc gamma receptor. Fc gamma receptors bind to IgG, while Fc alpha receptors bind to IgA. So if we think that IgA that binds to Shigella OSP mediates protective neutrophil phagocytosis, if we deplete IgA, we should see a reduction in phagocytosis, and this is what we do. So we depleted IgA from our samples, and we repeated neutrophil phagocytosis experiment, and we see that in the absence of IgA, there is a reduction in neutrophil phagocytosis. We then went further and decided to also look at monocytes because we know that they express Fc alpha receptor as well. And we see the same effect where when we deplete IgA from our samples, we see less monocyte phagocytosis mediated by OSP-specific antibodies. We then go ahead and look at other functions of neutrophils because they do so much more than phagocytose. They are actually very interesting cells. And so we look at granule secretion. These are bactericidal granules that neutrophils can secrete to kill bacteria and their surroundings. And again, we see that without IgA, there's a marked reduction of bactericidal granules secreted by neutrophils. And finally, we look at production of ROS, reactive oxygen species. And in the absence of IgA, there is a marked reduction of production of ROS. The last piece of the puzzle here was to go back to these resistant individuals and say, okay, we know that at some point later on, they did get infected with Shigella. So if we do believe that these OSP-specific neutrophil activating antibodies are protective, we should see that they lose them right before they get infected. And since we did collect samples over time, we were able to see, to compare baseline sample and the sample right before they get infected. And so this is what we did, and I'm showing you this here in green. This is how they entered the camp, and in blue, this is how they presented right before they get infected. And you can see that they lose these OSP-specific Fc alpha receptor binding antibodies. They lose neutrophil activation by these antibodies and also monocyte activation. And so if you lost me somewhere in the middle there, what I've shown you is this model of how IgA that binds to Shigella, or specifically to OSP on the surface of Shigella, can bind to Fc alpha receptor on the surface of neutrophils and activate them to promote bactericidal activities like granule secretion, phagocytosis, ROS production. And I only had time to show you this one little snippet, but we found in other cohorts, we found other mechanisms of protection. And now the next step would be going from these antibody profiling, where we take a bunch of human samples and just profile them unbiasedly, to ask very specific questions about the mechanisms of how the Fc fraction of antibodies actually protects against infection. So we will use an Fc engineering approach where we use the same Fab on the backbone of many different Fcs to then mechanistically dissect how antibodies actually protect against Shigella infection. And we can use these antibodies in many different assays. For example, things like I've shown you now, in vitro and ex vivo infection assays, but also infection of 3D culture models and in vivo infection models. And then we can always, or should always, go back to human samples and validate our findings, and then hopefully find other models of protection. And just to summarize, I've started with these very canonical ways that antibody Fc activates cells and complement, and I've shown you less canonical pathways that we found, like neutrophil degranulation and ROS production. Many other mechanisms were shown recently, like metabolic rewiring of cells via Fc receptor interactions, extracellular trap formation, and who knows what else we can find using these novel tools. And the last thing I want to do is just thank all of our collaborators, the study participants. Thank you, especially if you're in Israel and it's so late and you're still awake and listening to me. I'm happy to take questions. And if you don't have a question right now, just email me. Always happy to chat. Thank you.
Thank you very much, Biana. Any questions from the audience?
Okay, I can have one or two. One thing I want to ask is, do you see difference in neutrophil apoptosis when you between susceptible and non-susceptibles? And also, is there a specific subtype of IgA, like the dimers or the monomers? Can you tell?
Yeah, two great questions. So just to make sure that I didn't have enough time to really describe the system, but the neutrophils we use are from healthy donors. we don't have neutrophils from the patients. So we don't actually know what happens in the patients. Maybe someday, or maybe when we go into mice, we can actually ask these questions. Neutrophils love to die. So they definitely will probably perform apoptosis in response to this activation. But we usually look at live cells, so I can't really talk about that too much. The other question was dimeric versus monomeric IgA. Super interesting question that we're definitely going to ask using the monoclonal antibody system. So when we express IgA, we can make it dimeric or non-dimeric. In this case, these were serum samples, so I would assume that the IgA we're looking at is monomeric, because this is what we usually find in serum. But what I do know, at least from literature, is that binding to Fc alpha receptor should, at least what the literature says, is it should not be affected by dimerization, because the binding domain to Fc alpha receptor is exposed in the same way in the dimer and in the monomer. But it can change other things, like the way it binds to the bacteria, right? So it's an open question. It's very interesting, and we're definitely going to address that with the monoclonal system.
Thank you.
For interest of time, only one more question. So Abigail.
Thanks. Sorry, I wasn't allowed to unmute myself. Thank you. So maybe I'm a bit naive, but based on your data now, you now go back to the attempts at vaccine development and have a clue on to why they didn't work or how to make it better?
Yeah, how much time do we have? So I think the main thing is that, each failed attempt is its own story, right? Some failed attempts are because the vaccine is not immunogenic enough, so it just doesn't induce a response at all. And there it's really hard to know what went wrong. Other vaccines are really immunogenic, but not safe. And this also has happened before, where they tried to use whole bacteria vaccine, and it was just not safe enough. So it induced disease in some individuals. But I think the main, what I and we are trying to push for is, when we do test vaccines, usually people just look at IgA, IgG levels and serum, and that's it. If we did that for this cohort, we would see nothing, because there is no difference between resistant and susceptible individuals in their levels of IgA. They are the same. The difference we see is in binding to Fc alpha receptor. So what we are offering or suggesting or pushing for is looking beyond just IgA levels, IgG levels, but also looking at binding to Fc receptors and activation of innate immune cells as a protective feature of vaccine responses. And it is harder to do because it's not a simple ELISA, but it's not that hard. And if we do that early on, we can know before we go into children in sub-Saharan Africa and fail there, we can probably see vaccines that are potentially protective already when we start looking at phase 1, phase 2, and try to tweak that. But I mean, the question you ask is kind of like the holy grail of how do we make good vaccines, right?
Okay, thank you very much.
Thank you so much.
Thank you very much, Biana.